So in the previous lecture segment, we solved an example 2-point mapping problem, and we also built a composite map based on genetic distances that were provided from 2-point mapping type experiments. Now we move on to another kind of problem, 3-point mapping, but really 3-point mapping is where we're going to map 3 genes on a chromosome, and we're going to determine the relative distances between the genes. And it's really just two-point mapping problems that are being solved simultaneously. But since the map distances are smaller, we eliminate or at least minimize the problem of the 2-point map because we won't have as many undetected double crossovers between 2 points. So in any case, it's a slightly more accurate way of doing things. And also you get 3 genes on the map, not just 2. So just to review, in any genetic mapping cross, one parent is heterozygous for all the genes, all the loci under consideration. So in this case, it's going to be a triple heterozygote parent, and then the other parent is completely recessive for all the genes, and that's the mapping test cross. The phenotype of the offspring must reflect genotype, so we only deal with alleles that are fully dominant and fully recessive, so very straightforward genotype and phenotype correlations. And then you have to have a large number of offspring in order to get the most accurate measurements, the most accurate estimates of the distance measurements. And this is why humans, we have tried to calculate genetic distances between genes in humans, but it's hard to get enough data for obvious reasons. Okay, so these are the steps, the basic steps for 3-point mapping. And initially it looks like a lot, it's just a lot of words. And so we're going to go through each step, and I'm going to walk you through an example problem, and then we'll solve another problem. And you'll get to see a couple different ways that this can look. So I'm just going to start, and then I'll highlight each step as we go along. All right, so let's say you have a practice problem. So what's given is you have to have a triple heterozygote female cross to test cross males. That would mean the males are completely recessive. And the following progeny were counted. Now, really what they're giving you here is the alleles from the mother. You guys probably remember that for every individual offspring, of course, there would be a little x, a little y, a little z from the father, because he's recessive on all 3. So what happens is it just starts to be visually confusing. And so I don't write a lot of the times, well, when I write a problem, I don't write the chromosome from the father or from the test cross parent in either case. So you can see it's the same every time. So that's why I just don't even bother. It's assumed that you know that there have to be 2 copies of each chromosome. And we're really focusing on the chromosome that came from the mother, the one that came from the egg in this problem. And sometimes I'll give you a problem where you have to read the phenotypes and convert it into genotypes, and we'll practice doing that as well. But in this case, I've given you the genotypes, the allele combinations of the chromosome that came from the mother. And so the first step I give you on my list of steps is total the number of offspring. So I made this problem add up to a nice round 1,000, but you do need to double check that. And so 1,000 offspring total. Then the next step, really what we're doing next is we're trying to determine the order of the genes on the chromosome, the actual order of the genes on the chromosome. When you record the data, you just write the combination of alleles, but that doesn't mean you know how the alleles are actually laid out on the chromosome. So you should not assume that just because the alleles are written in a certain way, that means that is the order on the chromosome. It's just the way, just randomly how they were written down when we were counting the offspring. So you have to determine the order of the genes. Now, there's really no left or right, but there is a gene that's in the middle. What I'm saying is if the gene order is A, B, C, or A over here, and then B, and then C, either way, B is in the middle in that example. So it doesn't matter. You can rotate the chromosome, but the one that's in the middle will always be in the middle no matter how you rotate the chromosome. So we're really trying to figure out which gene is in the middle of the sequence of 3. And in order to do that, we're going to follow these steps. You're going to identify the parental allele combination groups and the double crossover or double recombinant groups. And you do that surely by numbers. So you'll recall that the parentals are the most common. So I circled the 315 and the 325. And the DCOs are the least common. So I circled the 13 and the 15. And so I've identified the most common and the least common. And I do label them like this because they could appear anywhere on the list. There's going to be 8 groups of offspring, and 2 of them are parental and 2 of them are double crossover groups. So we have to figure out what gene's in the middle and then rewrite the genes in the correct order. There's a couple ways to think about this. One thing I would say is, let's say that you had, for example, your parental, and I'm going to use different letters. Let's say your parental was big A, big B, big C, little a, little b, little c. Let's say that is the right order, and let's say that was your mother's 2 chromosomes. And let's say there was a double crossover, so a crossover here and here. What would happen is, if there was a double crossover, then you would have chromosomes that would be big A, little b, big C, and little a, big B, little c. And so as you can see, when there's a double crossover, the gene that's in the middle is flipped compared to just the straight parental allele combinations. So on the parental, it'd be big A, big B, big C, but on the double crossover, it'd be big A, little b, big C. So that little b is the one that's not matching up with the parental. And if you look at the other chromosome, little a, little b, little c on the parental, and on the double crossover one, little a, big B, little c. So again, the B is the one. So if you flip the B's back, if you look at your double crossover chromosomes and you reverse the B's, you put the big B back up here and the little b back down, you'll recreate the parentals. So you can look at it that way, or you can look at it, if I look at these 2 chromosomes, the A and the C are the same, but the B is the one that's different. And when I compare these 2, the A and the C are the same, but the B is the one that's different. So there's a couple ways to look at this, and we're going to practice figuring out what gene's in the middle. And so let's look at the actual problem that I've given you here. So your parentals are little x, little y, big Z, and big X, big Y, little z. And we're going to compare those to the DCOs, which are little x, little y, little z, and big X, big Y, big Z. And this one is actually pretty easy to see that it's the Z. If we take the Z's on the DCO chromosomes and flip them, and I put the big Z over here and the little z over here, that recreates what I have in the parentals. And so what you have is the Z should be in the middle. So when we rewrite the parental genotype, it doesn't matter which chromosome I list on top and which one I list on bottom, but my parentals with the correct order would go little x, big Z, little y, and then big X, little z, big Y. Now, it does not matter if the Y and the Z are switched, but most people would just keep as much of it the same as what was given in the problem and just make the minimum amount of changes in order to create the right gene order. So this is kind of the hardest part, in my opinion, is figuring out the correct gene order. And so that will take, don't worry if you're not 100% understanding it right now, because we're going to keep working on that. But Once you have the genes in the correct order and you know the mother's 2 chromosomes and how they're laid out, what we're going to do is figure out the genetic distance between each pair of genes, so each segment of the map. So if our mother's genotype is given here, then segment 1 is what I'm going to call the distance between X to Z. That's segment 1. And segment 2 is going to be the distance from Z to Y. So you're only going to solve each segment, the distance for each segment by itself. And that's why I say this is really a two-point mapping problems solved at the same time, simultaneously. So the distance from X to Z, and then the distance from Z to Y. So it's going to be a segment, what I call our segment 1 calculation and our segment 2 calculation. So I'm going to break here, and then we'll start another lecture part after this. Oops.